You may be familiar with Sir Alexander Fleming, the British scientist credited with the lucky surprise discovery of the first antibiotic, penicillin. His story is a firm favourite of high school biology teachers. A brilliant but untidy academic whose abandoned cultures of bacteria became contaminated. From all my experience in the lab, I've learned one thing: contamination is not good. For many scientists, it's disastrous and can ruin months of work. Thankfully, for Fleming, the invasive mold species had the opposite effect, because around the mold colony, an invisible line had formed, separating it from the surrounding bacteria. The mold had created an impenetrable zone of clearance where no bacteria can grow. The mold, whose official name is Penicillium notatum, had released penicillin, an antimicrobial toxin, literally a toxin against little life, which had killed off the tinier bacteria to reduce competition for food. After all, if there are fewer bacteria in culture, there is more food for the mold. A quick side note: antibiotics specifically kill bacteria, whereas antimicrobials refer to drugs that can kill any type of microorganism, like viruses and fungi. But let's go back to penicillin. To understand how it kills bacterial cells, we first need to take a closer look at bacteria. There's a vast range of different types, from acid eaters to glow-in-the-dark ones. But from the point of view of an antibiotic, all bacteria fall into one of two important classes. The first is gram-positive bacteria. These guys have a tough, thick cell wall made out of half protein, half sugar material called peptidoglycan, which is wrapped over the bacterial cell membrane. The other kind are gram-negative bacteria, which have a thinner cell wall sandwiched between an inner and outer membrane. The outer membrane has special channels called porins, which antibiotics take advantage of to enter these kinds of bacteria. Whether a bacterium is gram-positive or gram-negative affects its cell wall and in turn determines what can be brought into the cell and what its metabolism looks like. Antibiotics target processes that are specific to the type of bacteria. Let's use penicillin as an example. In a peptidoglycan network, separate strands of sugar are cross-linked together to increase the cell wall strength. For this to happen, a special protein called PBP has to bind to each sugar unit to allow cross-links to form. A part of the penicillin molecule binds to PBP and prevents cross-link formation, so cell wall synthesis is interrupted. Bacterial cells have their DNA and cytoplasms shoved in like clothes in a messy closet. Without the wall to hold its contents in place, they come tumbling out in a process known as lysis. If we look at this process under a microscope, with the cell wall stained a dark colour, we can see that by inhibiting cell wall synthesis, when the bacterial cell grows, the cell wall just can't keep up, and so the cell explodes. Because penicillin targets a molecule only found in bacterial cells with thick peptidoglycan walls, it cannot kill gram-negative bacteria such as E. coli and H. pylori. This also explains why antibiotics can't hurt our own cells. If we want to kill gram-negative bacteria, we need a different approach. Looking at the structure of H. pylori's outer membrane, we can see that it's made up of negatively charged fat molecules. So positively charged antibiotic drugs can interact with the membrane's molecules and create holes in the cell membrane, which weaken the bacteria's defenses, allowing the antibiotic to squeeze past and interfere with cell metabolism, stopping the bacteria from growing and releasing harmful toxins. But bacteria are not going down without a fight. Random mutations in individual bacterial cells can make the whole population more resistant to antibiotics. There are many mechanisms of resistance. Gram-negative bacteria can reduce their number of porins and change their cell's negative charge to a positive one to prevent antibiotics from getting in. Both types of bacterial cell can also develop efflux pumps to remove any antibiotics that are already in the cell. Resistant bacteria can even neutralize antibiotics by releasing toxins. Bacterial resistance doesn't take multiple generations to come about. Through horizontal gene transfer, one bacteria which has resistance to an antibiotic can transfer a little bit of its DNA to its neighbors to quickly build up a resistant population. Misuse of antibiotics can turn a normal bacterial population into a resistant one very quickly, and resistant bacteria are much harder to treat. Let's take a look at this process under the microscope in a population of Streptococci bacteria. Misusing or not completing a course of antibiotics can make it very difficult to deal with infections as bacteria become more and more resistant. But it can also have a great effect on the symbiotic friendly bacteria of your gut, which can cause issues with digestion and even lead to chronic inflammation. Antibiotic resistance is not a new concept. Alexander Fleming himself predicted it back in the early 1900s. And yet today, with socioeconomic issues and industry attitudes towards such drugs, resistance is now on the rise. In low-income countries, access to effective diagnostic tests might be limited, which means that the wrong antimicrobial might be prescribed for an infection, increasing the risk of resistance. Misuse of antibiotics as a preventative medicine is very common in livestock industries. An increase in meat consumption has made this problem even worse. By introducing policies which ban certain classes of antibiotics from being used in livestock industries, we've seen a good reduction in antimicrobial resistance cases. Another aspect to tackling antibiotic resistance is incentivizing drug companies to invest in making more antibiotics. Now is a really important time for research and development into antibiotic resistance. One of the most amazing studies I read recently involved bioengineering resistant bacteria to mop up excess antibiotics in livestock. I'll link to it in the description below. Tackling antibiotic resistance involves policy making and industry cooperation, but here's what you can do to help. Don't insist on taking antibiotics when you visit the GP, and if you are prescribed them, remember to take the full course even when you start to feel better. This applies to your pets too, when you take them to the vet. And remember, whilst it's great to share cookies and my YouTube videos, it's better not to share leftover antibiotics. In this video, we looked at how antibiotics work against two major types of bacteria, and we saw how bacterial cells can gain resistance to antibiotics too. If you enjoyed this video, please let me know in the comments below, and make sure to subscribe for more weekly science content.